Welcome to our deep dive into genomic editing techniques. In this video, we'll explore how scientists are rewriting the code of life itself. Genomic editing at its core involves making precise changes to the DNA of living organisms. This revolutionary field has opened up new possibilities in medicine, agriculture, and beyond. From curing genetic diseases to enhancing crop yields, the potential applications are vast. But how do these techniques actually work? Let's embark on a journey through the most important methods, their principles, and their real-world impact. First, let's understand what genomic editing really means and why it's so significant in today's world. Genomic editing refers to a collection of technologies that allow scientists to change an organism's DNA. These changes can involve adding, removing, or altering genetic material at particular locations in the genome. The goal is to correct genetic defects, treat and prevent the spread of diseases, and improve biological research. Genomic editing is a precise process, often compared to editing text in a document, except here, the text is the genetic code that determines how living things function. Let's look at the main tools that make this possible. One of the most groundbreaking genomic editing tools is CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Discovered in bacteria, CRISPR acts like molecular scissors, allowing scientists to cut DNA at specific spots. This technology has revolutionized genetic engineering due to its accuracy, efficiency, and relative simplicity. With CRISPR, researchers can target genes with unprecedented precision, making it easier to study gene function and develop new therapies. Let's see how CRISPR works in practice. CRISPR technology uses a guide RNA to find a specific DNA sequence. Once the target is located, the Cas9 enzyme cuts the DNA at that exact spot. The cell then tries to repair the break, and scientists can use this opportunity to add or remove genetic material. This method is not only fast but also highly customizable, allowing for a wide range of genetic modifications. The simplicity and versatility of CRISPR have made it the go-to tool for many geneticists around the world. For CRISPR, zinc finger nucleases, or ZFNs, were among the first tools used for targeted genomic editing. ZFNs are engineered proteins that bind to specific DNA sequences and create double-strand breaks. These breaks prompt the cell to repair the DNA, which can result in the insertion or deletion of genetic material. While effective, ZFNs are more complex to design and less flexible than CRISPR, but they pave the way for modern gene editing. Zinc finger nucleases combine a DNA binding domain, which recognizes specific DNA sequences, with a DNA cutting domain. When two ZFNs bind to adjacent DNA sites, they create a double-strand break. The cell's repair mechanisms then kick in, allowing scientists to introduce changes at the break site. Although powerful, designing ZFNs for each new target is labor-intensive and requires significant expertise. TALENs, or transcription activator-like effector nucleases, are another class of genome editing tools. Like ZFNs, TALENs use engineered proteins to target specific DNA sequences and induce double-strand breaks. However, TALENs are easier to design for new targets and offer greater specificity. They have been used in a variety of applications, from basic research to developing disease-resistant crops. TALENs consist of a DNA-binding domain derived from plant pathogens and a DNA-cutting domain. By customizing the DNA binding domain, scientists can target virtually any DNA sequence. When two TALENs bind to adjacent sites, they create a double-strand break, enabling precise genetic modifications. TALENs have proven especially useful in organisms where CRISPR is less effective. Eggonucleases are naturally occurring enzymes that recognize and cut long DNA sequences. Their high specificity makes them valuable for certain genome editing tasks. However, their use is limited by the difficulty of engineering them to target new DNA sequences. Despite this, meganucleases have contributed to the development of more flexible editing tools. Base editing is a newer technique that allows scientists to change a single DNA base without cutting the DNA strand. This method uses modified enzymes to directly convert one base into another, offering a more precise and less disruptive way to correct genetic mutations. Base editing is particularly promising for treating diseases caused by point mutations. Prime editing is an advanced technique that combines aspects of CRISPR and reverse transcription. It enables precise insertions, deletions, and base changes without causing double-strand breaks. Prime editing uses a modified Cas9 enzyme and a guide RNA to direct the desired edit, making it one of the most versatile tools available today. Gene knockout is a technique where a specific gene is completely disabled or removed. This allows scientists to study the function of that gene by observing what happens when it's missing. Knockout experiments have been crucial in understanding gene roles in development, disease, and physiology. Gene knock-in is the process of inserting a new gene or DNA sequence into a specific location in the genome. This technique is used to introduce beneficial traits, study gene regulation, or create disease models. Knock-in methods are essential for advancing genetic therapies and agricultural improvements. Homology-directed repair is a natural DNA repair process that scientists harness for precise genome editing. When a double-strand break occurs, a DNA template with the desired sequence is provided, and the cell uses this template to repair the break. HDR enables the insertion of new genetic material at specific sites, making it a key tool for knock-in experiments. Non-homologous end joining is another DNA repair mechanism used in genome editing. Unlike HDR, NHEJ simply joins the broken DNA ends together, often resulting in small insertions or deletions. This process is less precise but is commonly used for gene knockout experiments due to its efficiency. Genomic editing has transformed medicine by enabling the development of gene therapies for inherited diseases. Conditions like sickle cell anemia, cystic fibrosis, and certain cancers are now being targeted with precision treatments. By correcting faulty genes, these therapies offer hope for cures that were once unimaginable. In agriculture, genomic editing is used to create crops that are more resistant to pests, diseases, and environmental stresses. Scientists can enhance nutritional content, improve yield, and reduce the need for chemical pesticides. These advancements contribute to food security and sustainable farming practices worldwide. Genomic editing is also applied to animals, enabling the creation of disease-resistant livestock and the study of genetic diseases. These techniques can improve animal welfare, enhance productivity, and support biomedical research by creating animal models for human diseases. With great power comes great responsibility. Genomic editing raises important ethical questions, especially regarding human embryos and germline editing. Concerns include unintended consequences, genetic inequality, and the potential for misuse. Ongoing dialogue among scientists, ethicists, and the public is essential to guide responsible use of these technologies. Governments and international organizations are developing regulations to ensure the safe and ethical use of genomic editing. These rules address issues like safety, consent, and transparency. Effective oversight helps build public trust and ensures that the benefits of genomic editing are realized without compromising ethical standards. A crucial aspect of genomic editing is delivering the editing tools into cells. Common delivery methods include viral vectors, nanoparticles, and physical techniques like electroporation. The choice of method depends on the target organism, tissue, and the specific editing tool being used. One challenge in genomic editing is off-target effects, where unintended parts of the genome are altered. Scientists are continually improving the specificity of editing tools to minimize these risks. Careful design, testing, and monitoring are essential to ensure safe and effective outcomes. Genomic editing can target either somatic cells, which are non-reproductive, or germline cells, which give rise to offspring. Somatic editing affects only the treated individual, while germline editing can be passed to future generations. This distinction has significant ethical and regulatory implications. Gene drives are a powerful application of genomic editing that can spread specific genetic traits through populations. By biasing inheritance, gene drives can help control pests or disease vectors like mosquitoes. However, their use raises ecological and ethical concerns that must be carefully considered. Epigenome editing involves modifying the chemical markers on DNA rather than the DNA sequence itself. This can turn genes on or off without altering the underlying code. Epigenome editing holds promise for treating diseases linked to gene expression, such as cancer and neurological disorders. RNA editing is another innovative approach, focusing on modifying RNA molecules after they are transcribed from DNA. This allows for temporary and reversible changes, offering a flexible way to regulate gene function without altering the genome itself. Synthetic biology combines genomic editing with engineering principles to design and construct new biological parts, devices, and systems. This interdisciplinary field is driving innovations in medicine, energy, and environmental sustainability by creating organisms with novel functions. Genomic editing is paving the way for personalized medicine, where treatments are tailored to an individual's genetic makeup. By understanding and modifying specific genes, doctors can develop therapies that are more effective and have fewer side effects. There have already been remarkable successes in gene therapy thanks to genomic editing. Patients with previously untreatable conditions have experienced significant improvements, and some have even been cured. These stories highlight the transformative power of these technologies. Despite its promise, genomic editing faces several challenges. These include technical hurdles such as delivery and specificity, as well as social and ethical concerns. Continued research and responsible oversight are needed to address these issues and unlock the full potential of these technologies. The future of genomic editing is bright, with ongoing advances promising even greater precision and broader applications. Researchers are developing new tools and refining existing ones, aiming to make genomic editing safer, more efficient, and accessible to more people around the world. Public perception of genomic editing varies widely, influenced by cultural, ethical, and scientific factors. Education and transparent communication are key to fostering informed discussions and building trust in these powerful technologies. Global collaboration is essential for advancing genomic editing responsibly. Scientists, policymakers, and organizations from different countries are working together to share knowledge, set standards, and address shared challenges in this rapidly evolving field. As genomic editing becomes more widespread, education and training for scientists, healthcare professionals, and the public are increasingly important. Comprehensive programs ensure that these technologies are used safely and effectively, maximizing their benefits for society. Bioinformatics plays a crucial role in genomic editing by helping scientists analyze genetic data, design editing tools, and predict outcomes. Advanced computational methods enable more precise targeting and reduce the risk of off-target effects. CRISPR technology is not limited to editing DNA. New variants are being developed to target RNA and regulate gene expression, expanding the range of possible applications. These innovations are opening up new frontiers in biotechnology and medicine. Genomic editing is being explored for environmental applications such as restoring endangered species, controlling invasive populations, and cleaning up pollution. These efforts demonstrate the versatility and potential of these technologies to address global challenges. The cost of genomic editing has decreased significantly, making these technologies more accessible to researchers and clinicians worldwide. Continued innovation and investment are needed to ensure that the benefits reach all communities, not just a privileged few. As genomic editing technologies advance, questions about intellectual property and patent rights have become more prominent. Clear guidelines and fair practices are necessary to encourage innovation while ensuring broad access to these life-changing tools. The rise of the DIY biology movement has made genomic editing tools available to citizen scientists and hobbyists. While this democratizes science, it also raises concerns about safety, oversight, and ethical use outside traditional research settings. CRISPR-based diagnostics are emerging as powerful tools for detecting diseases quickly and accurately. These tests can identify pathogens, genetic mutations, and even cancer markers, making them valuable in both clinical and field settings. Editing the genomes of stem cells opens up possibilities for regenerative medicine. Scientists can create healthy tissues and organs for transplantation, offering hope for patients with conditions that currently have no cure. Genomic editing is being used in public health initiatives such as controlling mosquito populations to combat malaria. These efforts demonstrate how gene editing can have a direct impact on global health challenges. Artificial intelligence is increasingly being used to design, optimize, and predict the outcomes of genomic editing experiments. AI accelerates discovery, improves accuracy, and helps scientists navigate the complexity of the genome. Genomic editing techniques have transformed our ability to understand and shape the living world. As the technology continues to evolve, it holds the promise of curing diseases, improving food security, and addressing environmental challenges. However, responsible use, ethical oversight, and global collaboration are essential to ensure that these powerful tools benefit all of humanity. Thank you for joining us on this journey through the world of genomic editing.